The third type of macromolecule that we will discuss is proteins. Proteins are considered to be the most diverse macromolecule. Proteins are important in the cell because they comprise over 50% of the dry weight of cells. Polypeptides are considered to be the polymer form of amino acids. So the monomer unit of proteins is going to be amino acids. They're going to be joined together to form a polypeptide. A protein is considered to be a polypeptide chain, or more than one polypeptide chain put together. They will be folded in various ways to form a functional conformation. Whatever shape the protein takes will be very related to their overall function. In proteins, shape is very important in the overall function of the protein. Let's take a look at the monomer units involved and how these amino acids get connected together to form a polypeptide chain. If you recall from our earlier presentation on functional groups, each amino acid has an amino functional group as well as a carboxyl functional group. They also contain a unique side chain. If you take a look at the diagram to the bottom right, you will see that you have a carbon that's attached to a hydrogen and also an R group. This R group refers to the side chain involved, and it is going to be unique for each amino acid. To the right of that carbon, you have a carboxyl group, which is a C double bond O bond OH. You can see in the diagram below that this OH group is converted to an O minus. This is because at biological pH, remember, this functional group will be ionized. To the left of the carbon, you will find it attached to an amino group, specifically an N plus H3. Remember that once again at biological pH, the amino group is going to become ionized to form NH3+. The sequence of an amino acid chain will dictate the shape and function of the protein. There are 20 naturally occurring amino acids, and these are all going to be important in forming proteins. Let's take a look at how these amino acids are joined together. Once again, we are going to have what is termed a dehydration reaction. We are going to remove a molecule of water to join these amino acids together. In this case, however, we are going to form a peptide bond. There are three main categories of side chains for these amino acids. Amino acids can be classified as nonpolar or hydrophobic, polar or hydrophilic, or charged, either acidic or basic. This means that there will be regions within a protein that will correspond to the properties of the amino acids that are present. This leads to the uniqueness of the proteins. Once again, these amino acids are going to be held together by a peptide bond. The picture below depicts how that peptide bond is formed. It is formed from the carboxylic acid group of one amino acid interacting with the amino group of an adjacent one. Once you have a dehydration reaction and that water molecule is removed, you have the formation of that peptide bond, which results when that carbon from the carboxyl group is bound to the nitrogen of the adjacent amino group. Now we are going to take a look at all the different types of amino acids. I do not expect you to know the individual side chains. I do, however, expect that if I am giving you an amino acid, you should be able to tell me the properties of that amino acid. And I will show you what I mean as I take you through these slides. These are considered to be your nonpolar amino acids. Remember, when something is nonpolar, that means that the electrons are going to be shared equally between those atoms. If we take a look at all of the amino acids below, what I would like you to note is that most of them simply consist of carbon, hydrogen interacting with each other. There's nothing out of the ordinary with any of these amino acids. They might have a carbon in the ring form, but you don't see any charges on any of these amino acids. The one I want to point out, if we take a look at methionine. So methionine does have that sulfur atom in between those two carbons. So if you remember on the periodic table, sulfur is very close to oxygen. Oxygen has a very high electronegativity. Note that we do not see oxygen in any of these side chains. We do see sulfur, which once again has a bit higher electronegativity. But when you have sulfur in between two carbons, what happens is those electrons are going to overall be shared equally amongst that carbon to sulfur to carbon bond. As a result, we consider methionine to be nonpolar. If an amino acid is nonpolar, these are all going to be classified as hydrophobic. So these amino acids are not going to interact very well with water. Now let's take a look at the polar amino acids. All of the amino acids below have something in common. Aside from cysteine, all of the amino acids below have oxygen present. If you remember, oxygen is considered to be highly electronegative, which means that it's going to be pulling electrons closer to it. As a result, this gives polarity to the side chains. So these are considered to be polar. They're also considered to be hydrophilic. Now let's consider cysteine. Cysteine has sulfur. Like I stated in the earlier slide, sulfur is considered to be highly electronegative as well. When it is present between two carbons, however, that electronegativity leads to equal sharing of electrons. But if we take a look at cysteine, we can see that the sulfur or sulfhydryl group is at the end of that side chain. That means that, like oxygen, it is also going to be pulling the electrons closer to it, and as a result, it would give polarity to that side chain. So when in doubt, when you see oxygen present, think polarity. When you see sulfur present, you can think that as well, unless it is placed in between two carbons, at which point it would then lead to equal sharing of electrons and it would be considered nonpolar. And finally, let's look at the last group of amino acids. These are the electrically charged amino acids. These will be easy to define because you should see positive or negative charges associated with the side chains. If you have the side chains being negatively charged, this would be considered acidic. Remember that C double bond O used to be C double bond OH, which is your carboxylic acid functional group. If you have positive charges and amino groups associated with your side chain, then these would be considered your basic amino acids. So once again, to recap, I do not expect you to know and memorize how to draw out all of the amino acids. What you should know is that every amino acid has a consistent base structure, which includes a carbon attached to an amino group, which is attached to a carboxyl group. So there's two important functional groups that are usually ionized at body pH, which means that the amino group is going to be in the form of NH3+, and the carboxyl group would be COO-. What I also expect you to know is that these amino acids can either be nonpolar if the electrons are shared equally, polar if there's the presence of oxygen in the side chain, or if you have sulfhydryl group at the end of the side chain. Or these amino acids can be electrically charged, acidic if they're negatively charged and contain the carboxyl group, or basic if they're positively charged and you can identify that amino group. Now let's take a look at peptide bond formation, so how these polypeptide chains are formed. If you haven't noted already, one thing that is common in the formation of all macromolecules is the importance of a dehydration reaction. So all of these macromolecules are going to be formed by removing water. In a peptide bond formation, this is going to occur in the ribosome. The ribosome is going to be responsible for catalyzing this reaction and joining multiple amino acids together. This is done in a chain reaction where you begin with one amino acid, you then attach another one to form a dipeptide, another amino acid will join to form a tripeptide, and eventually this will continue on to form your polypeptide chain. New amino acids are always added to the carboxyl group of the growing chain. So in amino acid 1, you will have your amino group to the left, your carbon with the hydrogen and with the side chain, and then you will have to the right your carboxyl group. This is going to be lined up with an adjacent amino acid, specifically with the amine group of amino acid number 2, A dehydration reaction is going to occur, a peptide bond will form, and then that would result in the carboxyl end being free to join with amino acid 3. That means that for every single protein produced, we have a beginning and we also have an end. At the beginning of the amino acid chain, you would have the amino group being exposed. This refers to the N-terminus of the polypeptide chain. As the polypeptide chain grows, the carboxyl group would then be exposed, and when you are finished your polypeptide chain, that last amino acid would have the free end being the C-terminus. So when we have a glycosidic bond, we can refer to that bond as 1,4, 1,2, depending on the carbons that we have. And when we have a polypeptide chain, we can refer to the ends of them as being either the N-terminus if they have the amino group, or the C-terminus if the carboxyl group is exposed. Proteins can have many different functions in the cell. By far, most of these proteins in the cell are going to be enzymes. Enzymes are very important in catalyzing many chemical reactions in the cell. Proteins can also be structural and provide support for the cell. Examples of this are collagen, elastin, keratin. They can also give a lot of flexibility to the cell and resist a lot of tensile forces. You can also have storage proteins, which act as storage forms of various types of amino acids. These are really important in egg development or in casein in breast milk. These provide growing offspring with many different types of amino acids that will then be incorporated into their own proteins. You can have proteins that are involved in transport, such as hemoglobin, which is a protein important in red blood cells that helps to transport oxygen. Proteins can also transport ions across the membrane, such as a proton pump. Proteins are also important in creating hormones and receptors, and an example of this would be the insulin and corresponding insulin receptor. These are both polypeptide-based, and as a result are classified as proteins. Proteins are also important in contracting and also in providing motor proteins. Actin and myosin are very important in performing your muscle contraction, where these fibers will lay on top of each other and they will move accordingly to contract the cell. And when multiple cells contract, you can contract an entire muscle in a coordinated response. Proteins are also very important in providing immune defense. They provide the antibodies that are necessary to fight various types of infection. An important aspect of enzymes is they are typically named for what they do. For instance, sucrase is an enzyme in the cell that is responsible for digesting sucrose molecules. An enzyme's shape is very important and is related directly to their function in the cell. Enzymes can be responsible for bringing products and also cofactors together in what we term the active site. The active site is the site of catalytic activity in an enzyme. It is at this site that the reaction actually occurs. Many enzymes can be considered a catalyst because they facilitate chemical reactions. They are not used up in the reaction, which means that they do not change the product or the reactant. They simply provide a space for this reaction to occur. They also speed up the rate of reaction and make the reaction more feasible to be carried out in the cell. They're not used up in the reaction, so they can be used in multiple reactions sequentially. Let's take a look at protein structure. There are 4 different levels of protein structure. What's important to note is that the primary, secondary, and tertiary level of a protein will be consistent. However, not all proteins achieve a quaternary or 4th level of structure. The primary structure of an amino acid simply refers to the amino acid sequence. This is your base polypeptide chain that consists of all of your amino acids joined together by the peptide bonds. You would see an N-terminus and a C-terminus within your polypeptide chain, designated in your primary structure. Everything in your protein will depend upon this primary structure. Secondary structure is when you have your polypeptide chain and it begins to take on a 3D shape in the cell. There's two main secondary structures that can be taken on by the polypeptide chain, specifically the alpha helix or the beta sheet. Most of these structures will be held into place by the hydrogen bonds between the backbone atoms. So once again, these hydrogen bonds that we saw important in how water interacts with other molecules as well as itself, hydrogen bonds become very important in maintaining the secondary structure of a protein. This is an example of what that secondary structure would look like, specifically the alpha helix versus the beta pleated sheet. You can see that the dotted lines refer to the hydrogen bonds that are responsible for holding these shapes in place. The tertiary structure of a protein occurs when you have multiple beta sheets and multiple alpha helices that are interacting with each other. At this point, there is a complex shape that the polypeptide will begin to form. This mainly involves interactions of the side chains once again, but now we're going to have more complex interactions. You're going to have important bonds such as disulfide bridges form. You're going to have hydrophobic interactions. You're also going to have van der Waals forces, either attractive or repulsive, that are going to come into play that will help a protein take on a tertiary structure. If you consider the secondary structure, the primary bond there is the hydrogen bond. And finally, the quaternary structure involves the interaction of multiple polypeptide chains to form one functional protein unit. This means that sometimes a protein will completely be able to take its shape and function at the tertiary structure. However, there are some proteins that are more complex, and if they involve multiple polypeptide chains, then a quaternary structure would need to be formed. Here is an example of tertiary versus quaternary structure. Once again, tertiary structure is simply when one polypeptide chain takes on that 3D shape with multiple alpha helices and beta sheets interacting with each other. Quaternary structure is formed when multiple polypeptide chains interact with each other. Another important concept to understand is the concept of protein folding. Folding refers to when the polypeptide chain starts acquiring its secondary and tertiary structure. The folding or shape of a protein is going to be determined by that primary polypeptide sequence. Some proteins are small and can actually fold on their own, but more complex proteins are going to require some assistance. This assistance comes in the forms of chaperonin proteins that basically grab onto the polypeptide chain to ensure that it is folding in the proper formation. When a protein encounters some extreme conditions, such as changes in pH or temperature, changes in salt concentration, this may disrupt the bonds that are present in the folded protein. This may cause the protein to unfold or denature. What is occurring in these extreme conditions is all of the important bonds, such as the disulfide bonds, the hydrogen bonds, the van der Waals forces, are being disrupted. As a result, the protein will unfold and maintain and go back to its primary state. When a protein is denatured, it no longer can function normally in the cell. This protein denaturation can sometimes be reversible, but sometimes it is permanent and the protein will never attain its tertiary or quaternary shape again. This image depicts the cycle between protein formation and denaturation. So we have a normal protein on the left. If it is exposed to extreme conditions, once again, those bonds would be disrupted and the protein would unfold and go back to the primary sequence. This brings us to our last important group of macromolecules, the nucleic acids. Nucleic acids are found in the nucleus. There are two different types of nucleic acids, DNA or RNA. It is important to note that there are base structural differences between these two nucleic acids. One of those structural differences has to do with the base sugar that the macromolecule is based on. In a DNA molecule, that base sugar is going to be deoxyribose, and in the RNA molecule, you are going to have it based off of the ribose sugar. Please take a look at the diagram to the right to see the difference between deoxyribose and ribose sugar. This difference lies in the number 2 carbon. Deoxyribose does not have the oxygen attached to it, whereas ribose has the oxygen and hydrogen in a hydroxyl group attached to the number 2 carbon. These two nucleic acids differ with respect to their role in the cell. DNA is the repository of genetic information, and RNA is involved in the expression of this genetic information. Both are considered to be polymers of the base unit, the nucleotide. Nucleotides are going to be held together by a phosphodiester linkage. So, to recap, we had glycosidic linkages in sugars, we had peptide bond linkages in your proteins, we had ester linkages in your fats, and here in the nucleic acids we have a phosphodiester linkage. Once again, how this linkage is formed is going to be similar. You're going to have a dehydration reaction that helps to form this linkage. Once again, if you want to break this linkage, then a rehydration reaction would have to occur. When you are talking about these nucleic acids and their role in the cell, it's important to remember the central dogma theory of molecular biology, where DNA acts as a template for genetic information. RNA will then be built off of that DNA template, and this will ultimately lead to the formation of a protein. Here we can see in the diagram below, this illustrates that concept. We have a double-stranded DNA, which acts as a template. RNA is going to be made from that DNA template. That will then be transported into the cytoplasm. Ribosomes will then come in to help express that information. So we can see here the linkage between nucleic acids and also between your proteins. It is important to know the base structure of a nucleotide. A nucleotide can be broken into 3 different parts. 1, the 5-carbon sugar that forms the base. Remember, this is going to be deoxyribose in DNA, or this sugar will be a ribose sugar if you are dealing with RNA. You will also have a nitrogenous base. That base can be A, C, G, or T, and it can also be U, which stands for uracil. Uracil is an important nitrogenous base that is only present in RNA. Thymine, or T, is an important nitrogenous base that is only found in DNA. However, if you take a look at A, adenine, G, guanine, C, cytosine, those nitrogenous bases are going to be common in both DNA and RNA. Another important component to a nucleotide would be the phosphate group. So the phosphate group will be attached to the 5-carbon of the sugar. These nucleotides can contain 1, 2, or 3 phosphate groups. If you recall, ATP was a nucleotide that had 3 phosphate groups, and we will see how important ATP becomes in the cell when we get to respiration. Nucleotides are polymerized or made by enzymes that are called polymerases. We'll have specific polymerases depending on what type of nucleic acid we're going to make. So DNA polymerase is involved in making DNA, whereas RNA polymerase will be involved in making a strand of RNA. It is important to note that just like we had a 1,4, 1,2 glycosidic linkage, and we also had an N-terminus or C-terminus of a protein, when we're dealing with nucleic acids, we can have a 5' end or a 3' end. These ends are going to refer to which carbon is exposed at that particular end of the strand. When we're connecting nucleotides together and when we are forming those phosphodiester bonds, you start off with the 1 nucleotide base. And as a result, that is going to have the 5-carbon attached to it, which you can see at the 5' end in the diagram to the right. When you are adding subsequent nucleotides, these are going to be added at the 3' end. You're going to have a phosphodiester bond form through a dehydration reaction, and you're going to have now 2 nucleotides tied together. DNA polymerase is going to come in and assist in catalyzing this reaction. When you get multiple nucleotides added together, they are also going to be added at the 3' end. So this means that at the end of this chain, you will have a 3' carbon that will be exposed. We call this the 3' end. There are some important characteristics to the DNA structure. DNA exists in the cell in a double helix. This means that there will be two strands joined together and they form a helical nature. These two strands are lined up in opposite directions, meaning that they run anti-parallel to each other. That means that when you have a 5' end of one strand, it is going to be paired up with the 3' end of an adjacent strand. Similarly, at the end of that, you would have a 3' end paired up with a 5' end. So because one strand runs from 5 to 3, the other strand runs from 3 to 5. This is what we refer to as anti-parallel strands. They will then form a double helix. This means that because of the interaction between the nitrogenous bases when they join up together, it will form a coiled or helical structure. Sometimes you will hear the DNA double helix referred to as a ladder. That means that the backbone of the DNA molecule would form the sides of the ladder, which would consist of the sugars and the phosphate groups and the phosphodiester bonds, whereas the interaction between the nitrogenous bases would be considered the rungs of the ladder. The bases of the nitrogenous strands join together with hydrogen bonds. These bases also pair up in a specific way. We call this the base pair rule. This is also called Chargaff's law and is consistent amongst all DNA molecules. This specifically states that when you have a cytosine, it will only pair with guanine. When you have adenine, it will only pair with thymine. Or if you're dealing with a RNA molecule, that would be pairing up with uracil. These nitrogenous bases are paired up in a consistent way, and they're paired up through the presence of hydrogen bonds. If we take a look at these hydrogen bonds, when you have adenine and thymine together, they are going to be joined with 2 hydrogen bonds, whereas G and C will be bound together with 3 hydrogen bonds. Once again, remember that these strands are going to be in anti-parallel directions, so one will be running from 5 to 3, and the other strand would be running from 3 to 5. This means that if we have a sequence of DNA, we can automatically determine what the complementary sequence of DNA should be. So an example, if we have 5' and that is followed by ACCGTGA 3', that means that we know that the corresponding strand should be as follows. This is because they follow the base pair rule and that they also run anti-parallel. So we know if we're dealing with a 5' end in one strand, that would be lining up with a corresponding 3' end. And that ends our topic of the biological macromolecules.